Alright, welcome back to this channel. In this video, we are going to be talking about the cell. The cell. And we're going to be covering both the animal and the plant cell. Now, I will enjoin you to sit tight because we're going to discuss a good number of things that you need to know, both for an average student and advanced, or would I say semi-advanced students, under the topic of cells. So do well to watch the video from the beginning to the end. Exactly, and drop any questions you have for me in the comment section. Okay, now we're going to be dealing with the introduction, the history of cells, cell theory, and the cell organelles. Okay, I'm going to be dwelling much more on the cell organelles. Now, the cell biology is a branch of biological sciences which deals with the study of structure, function, molecular organization, growth, reproduction, and genetics of the cell. That is cell biology. It deals with structure, function, that is function of the organelles, molecular organization, that is how the structures, the organelles in the cell are formed, the growth and reproduction of the cell, the genetics of the cell, that is how materials are passed on from one generation to the other. Now, cell biology mainly concerns itself with the study of structure of cell and function of specialized organelles. That's what it concerns itself with mostly. Okay. Now, a brief history about the cell. Now, the term cell was given by Robert Hooke, and that is in the year 1665, and he's called the father of cell, okay? And he recorded his finding on the cell in his book that he titled Micrographia. That was his first book on the cell, or primarily the first book on cell. And he named the structure that he observed in a cork. He called it cellular because he said they looked like small rooms. So the root word for cell is cellular, which means small rooms. Okay. So he did this in the mid-17th century to describe the structure of the cork. Okay, the cork is just the dried bark of the leaf of the tree. Okay. Now, Anton van Leeuwenhoek now was the first to record observations of bacterial cell. So he called them animalcules because the structure he saw, what he did was to modify the already existing microscope in order to be able to view smaller organisms such as protists and bacteria. And for this reason, he was called the father of microbiology. So he was called the father of microbiology. So don't forget, Robert Hooke, father of cell, Anton van Leeuwenhoek, who was forced to view the lower organisms that he called animalcules. He was called the father of microbiology, and he collected these structures from pond water and tooth scrapings. Okay, now in 1830s, the importance of cells were realized. Okay, so we'll see that in 1838 and 1839, two scientists, one was a German botanist called Martha Schleiden, observed that despite differences in tissue structure, all plant tissue were made of cell. Even though they had different structures, all plant tissues contained what? Cell. So he was a botanist. He said all plants were made up of cell. Then in 1839, following this, was Theodor Schwann, who realized that animals were also composed of what? Cell. That cell was the fundamental unit of the animals, okay, of the animal tissue primarily. So Schwann proposed the first two principles of the cell theory. Schwann was one that proposed the first two principles of the cell theory, and his principle was that all organisms consist of one or more cells. All organisms consist of one or more cells, and that the cell is the structural unit of life. The cell is the structural unit of life. The Schleiden-Schwann view of cell origin was less insightful. That is, cell could arise from non-cellular materials. Their view of the cell was that the cell could arise from non-cellular materials, okay? And this was not exactly true, okay? This was not exactly true. That was the only mistake in their proposition. However, this was the idea was corrected by Rudolf von Virchow. Rudolf von Virchow, and he was one that demonstrated that living cells could arise only from other living cells, the process known as biogenesis. Biogenesis, bio meaning life, and genesis, genia means origin, or to, genesis will mean to originate. Exactly. So biogenesis is the process by which other cells um, um, originate from cells, new cells originate from previously existing cells. Then abiogenesis is the theory, the opposite of that. So Rudolph proved that organisms did not exist from inanimate matter, but from animate matter, that is previously existing cells, previously living cells. Okay, so let's now discuss about the cell in full. The cell is the basic structural and functional unit of cellular organisms, except viruses. The cell is the basic structural and functional unit of cellular organisms. Now, the cell is from a Latin word that means small arm. Sorry, I said Greek. That was Latin, cellular. I'm sorry. Micrographia, I just remembered. Micrographia, Latin. Okay? So, the cell is a Latin word that means what? Small rooms. Small rooms. Okay? Now, A.G. Louis and P. Siekevitz in 1963 have defined cell as a unit of biological activity delimited by a semi-permeable membrane and capable of self-reproduction in a medium free of other living systems. So don't let this confuse you. Delimited by semi-permeable membrane means it is covered, that is, it is bound by a membrane, and that it is also capable of self-reproduction, and in a medium free of other living system. Okay? Now, the properties of the cell. Cells are generally highly complex and organized. Yes, the cell is the first level of organization of life. Do you understand? This is the first level of organization of life. However, we know that biology, if you have seen my previous video on introduction to cell biology, I have talked about how that level of organization of living things actually begin from non-living, evolutionary speaking. So we'll talk about atoms as the first level, then um, atoms now come together to form molecules. When after the molecules, atoms molecularize, then we now have molecules that are now aggregated together to form larger molecules, which are macromolecules. But you see, when these larger molecules are bound together, they form organelles. They are bound by a membrane that they form organelles. Now, these organelles now function together to form the cell. So it is these organelles that are, in fact, the word organized. Do you understand? Organelles. So these organelles function together to form the organized structure called the cell. Do you understand? So they are highly complex and organized. Now, the cell possesses a genetic program and the means to use it. They possess the genetic program embedded or encoded in the DNA and the means by which it is used or transferred from one generation to the other. The cells are capable of producing more of themselves. That is self-reproduction. The cell is self-reproducing. Okay? Now, the cells can acquire and utilize energy. The cells are capable of acquiring and utilizing energy. They manufacture energy using mitochondria. They store it in form of ATP in the same, and they are able to utilize it. They carry out a variety of chemical reactions. They carry out a variety of chemical reactions. Many chemical reactions take place in the cell. Okay. For example, one is there are just many. Even in the respiration, we talk about is a chemical reaction where glucose is broken down to give um, carbon dioxide and and um, carbon dioxide and water, and peroxisome when it peroxidizes, when it when it catalyzes the breakdown of peroxide. Just many chemical reactions take place in the cell. Then they also engage in mechanical activities. Yes, they engage in mechanical activities. For example, plant cell. For example, when they absorb water, they become turgid and they are able to stretch. They stretch. These are mechanical activities. Some cells actually move also. Such cells are flagellated, so and they are able to move around. Okay, they're also able to respond to stimulus. Yes, you see, they have the cell membrane that delimits the cell has studied within it the certain receptors that helps in the cell to cell communication. Is that okay? Cell responds to its environment, responds to the secretion of things within it and without it. Okay, so that is response to stimulus, and they are capable of self-regulation. Yes, the cell can regulate itself by the control center known as the nucleus. Is that okay? So the cell regulates itself, knows is capable of directing its own activity by the action of the nucleus. So that's why the cell is generally what termed as organized, very organized and complex. Okay. Now let's talk about the types of cell. We have the eukaryotic cell and the prokaryotic cell. And the eukaryotic cells are divided into the plant cell and the animal cell. Okay? And the prokaryotic cell are the archaea and the bacterial cells. Okay? Now let's talk about the differences between the prokaryotic cell and the eukaryotic cell. The prokaryotic cells are small, they are small in size, usually between 1 to 10 micrometers. And I've told you micro means times 10 to the power minus 6. Okay? So usually between 1 to 10 micrometers, that is the size of the prokaryotic cell. We can see the eukaryotic cells are large. Compared to this, they are about between 10 and 100 micrometers. That is way larger than the prokaryotic cells. So which means that a typically big prokaryotic cell, relatively big though, prokaryotic cell will need will have to, is still 10 times smaller than a big eukaryotic cell. All right. So then the prokaryotic cells are generally unicellular. They are generally unicellular, but the eukaryotic cell, they are generally multicellular. Is that okay? Even though there are some unicellular forms of eukaryotic cell, they are mostly multicellular. Okay? Now, the prokaryotic cells have single membrane and cell wall. They have a single membrane and cell wall. We have membrane bilayer. We have double membrane. Our plasma membrane is double. Is that okay? But we have no cell wall. These guys have, okay. They're actually cell walls for plant cell. I'm sorry. Inside, I said the eukaryotic cell are divided into both plant and animal cell, okay? Now, the plant cell have cell wall. The animal cells do not have cell wall, okay? So, but both of them have cell membrane. The plant and animal cell have cell membrane, which are both what? They are bilayers. That is, they are made of phospholipid bilayers. They are double membrane. Do you understand? They have double membrane, while the prokaryotes have single membrane and cell wall. Then the prokaryotes have no nucleus. In fact, what they have is a nucleoid. A nucleoid. And I've told you the word oid means false or like. So it means it is like that thing, but not exactly that thing. That is nucleoid. So it looks like the nucleus, but it's not the nucleus. It is basically the DNA, the chromatin material that is not bounded. So if it is bounded, it is only bounded in eukaryotic cell, but it's not bounded in the, it is not bounded by a membrane in the prokaryote. So because of that, we say it is not a nucleus. The nucleus is both the membrane that is surrounding the nucleus and the genetic material, the membrane that is surrounding the genetic material and the genetic material itself. That's what make up the nucleus. So these guys have no nucleus. Okay? Now, the prokaryotes have circular DNA, while the eukaryotes have linear DNA. So their DNA is circular. Is that okay? While we have a linear DNA. Our DNA has a double helix structure. Make sure you follow the biochemistry series because I'm going to be explaining a lot about this double helix structure as um, described by Watson Crick, the Watson Crick model. Okay, so these guys, prokaryotes, have no histones. They have no histones. That's why their chromatin material is loosely bound, is not protected, is not condensed and bound together by histone proteins. But these guys, eukaryotes, have histone proteins that help them to compress the size um, of the genetic material, the chromosome, such that it can be contained within the nucleus. The prokaryotic cells have ribosomes, okay, but the ribosomes are freely floating within the cytoplasm, okay. Now, the eukaryotes have ribosomes on the surface of the rough endoplasmic reticulum. Albeit there are some that are floating about in the cytoplasm, but most of the ribosomes in the eukaryotic cells are on the membrane. They are on the surface of the endoplasmic reticulum, the membrane of the endoplasmic reticulum. Okay, now the cell division by in these guys prokaryotes is by fission. Okay, but in eukaryotic cell is by mitotic division. Even though they look alike, but the mechanism that power both processes are different. Then they are direct both processes. Sorry. Then the cytoskeleton, it is absent. Okay, cytoskeleton is absent, yes, in prokaryotes, but it is present in eukaryotes. Okay, and the cytoskeleton that they are supposed to have is rather their flagella. So a prokaryotic cell will have flagella at the top, which it uses to move. Okay, and it is the structure of that flagella or the nature of that flagella is similar to the nature of our microtubules, cytoskeletons, microtubules in the plant cell, animal and plant cell. Then, example of the prokaryotes is the bacteria or rickettsia, and the example of the eukaryotes is the cells in fungi, plants, and animals. Okay. Now let's see the prokaryotic cell. Now you see the bacterial flagellum, Then you will see scattered within here is the ribosome, freely floating, non-membrane bound. And it is small. It is very small compared to what you have in the eukaryotic cell. Then there is the DNA nucleoid. Okay? Then there is also the plasma membrane, which is the cell membrane, just after the cytoplasm of this cell. Then you have the cell wall that covers the cell membrane. Then we have the capsule. The capsule now covers the entire membrane. The capsule is the one that is the outermost covering of the prokaryotic cell. So notice that the DNA of nucleoid is just scattered around. They're like unbounded. It's unbounded. It's free. Yes, that's the word. It's free inside the cell. Okay. Then we have the eukaryotic cell. the animal cell and the plant cell. As you can see here, you're going to see the content of the nucleus here. You're going to see the content of the nucleus. We have the nuclear envelope, the nucleoplasm, that is the fluid inside the nucleus, and the nucleolus. The nucleolus is just a portion of the nucleus that that provides or makes the structure of RNA, ribosomal RNA, for the production of ribosome. So basically, we say the nucleolus manufactures ribosome. This one also has the plant cell also has the four components that we see in the animal cell. Okay, but you see structurally they are different. Structurally they are different. This one is rectangular. This one is ovoid. Okay. Now you also see other organelles. Now you see chloroplast here that is not present in animal cell. You're also going to see the vacuole is large. Vacuole is large. While here the largest is the nucleus. The vacuole is so large that in some parts of the cell, of the plant cell, it pushes the nucleus to one side of the cell. But in animal cell, it is usually the nucleus is usually centrally placed, centrally located. Okay, you see what else? What else? You're also going to see this. There's also now. What does this guy also have that this one does not have? This one have plastids. Apart from the chloroplast, we also have the leucoplast and the chromoplast. All of these are not present in the animal cell. But one other thing that this guy has that this guy does not have is that this one has the centriole or centrosome. This one does not have the centriole. The centriole are in the centrosome, so they are not the same. The centrioles are in the centrosome. Okay, so the centrioles here have the primary function of for cell division. Okay, so centrioles are not in the plant cell. Okay, so this is another 2D diagram. This one shows the 3D diagram, the former one we saw. This one shows the 3D, the 2D rather. So here we have the mitochondria. You can see the cell wall, which is not present in these ones here, are the ones that are not present in the plant, in the animal cell. Okay, so you can see a small invagination here. This small invagination here depicts the outlet of a vesicle. A vesicle is trying to deliver something outside of the cell. How does it do that? What it does is it will merge with the membrane of the cell. It will merge itself. That's the vesicle. These vesicles are usually from the Golgi apparatus. So they will go and merge with the plasma membrane. They will form part of the plasma membrane and they will exude their content. Okay, so we have the cell mitochondria, we have the centriole, we have the chloroplast. Okay, these two are centriole is present in plant animal, chloroplast is present in plants. Then we have the ribosome, we have the endoplasmic reticulum, we have the nuclear membrane. The nuclear membrane is just the nuclear envelope that we saw before, it's the same. The nucleus, you can see the nucleus and you can see the nucleolus. Okay, then you can see the chromosome that are floating around inside the nucleus. Okay, then we have the vacuole. You can see the vacuole is very large, it's the largest organelle in the animal cell, in the plant, I'm sorry, in the plant cell. But here you see the vacuoles, our own vacuoles are very tiny, very tiny. Do you understand? These guys here, they are vacuoles, very small. Okay, so that's that. Then we have the Golgi apparatus. The Golgi apparatus. Hopefully, if I'm able to talk about the endosymbiotic theory here and the MIT, the membrane invagination theory or the endomembrane theory. I will so that you understand the relationship between the endoplasmic reticulum and the nuclear membrane and the plasma membrane and the Golgi apparatus, the smooth and rough endoplasmic reticulum, and of course the vesicles also. So you should understand the relationship between them. So I'm going to, it's possible I talk about it in this video. Okay, so you can see that the animal cell has flagella to a flagellum. Okay, so that's that. Then we have components of a cell. A cell is composed of hyaloplasm and various structures or cell organelles like cell wall, cell membrane, or plasma lemma. Plasma lemma is also called cell membrane or plasma membrane. So it has three names: plasma membrane, cell membrane, or plasma lemma. And the endoplasmic reticulum, which are two RER and SER, the ribosome, the Golgi apparatus or Golgi body, the lysosome, the spherosome, the chloroplast, the mitochondria, the centriole, and nucleus. So the cell organelles. Now, organelles means little organs. Organelles means what? Little organs. Little organs. Okay, a small structure in the cell that performs a specific function. Is a small structure in the cell that performs a specific function. Okay, and membrane-bound organelles are found only in eukaryotic cell. So we have cell organelles. There are membrane-bound ones, and there are membrane non-bound. That is, there are some that are non-membrane bound, and there are some that are membrane bound. The membrane bound are split into two. The membrane bound are split into two, which are the double membrane and the single membrane. The single membrane will be like, the example will be like the endoplasmic reticulum, while the double membrane will be the nucleus, the plasma membrane, the mitochondria, chloroplast. But here we have endoplasmic reticulum, Golgi apparatus, vesicles. Those guys are single membrane. You understand now? Okay? Even though I didn't include it. All right, so let's begin with the cytoplasm. In a cell, except the nucleus, is cytoplasm. Everything in the cell except the nucleus is what? The cytoplasm. Because it is the clear gelatin-like watery substance surrounding the organelles. Now let me show you some. The word cytoplasm is gotten from two words. Anytime you hear the word cyto, it means cell, okay? And you hear the word plasm. Plasm is from the word plasma. You know plasma in the blood, right? The fluid portion in which other cells float inside in the blood, yeah? Okay? So, and the plasma typically is contained of what? Water. In fact, the word plasma actually means from the Latin word mold. Do you understand? To mold, to form something. Do you understand? Like liquid like. Do you understand? Okay? So that is so we call it the cytoplasm. So it is the one, it is clear, it is gelatin-like, and it is composed of 90% water. And it is in it that every other organelles float. Okay? Then we have the organic and inorganic compounds in it at 10%. On 90%, it's actually in some textbooks you might see 80 to 90% water, and depending on the cell, actually, then 10% dissolved substances, inorganic and organic. All right. Now, so that's why when you see the structure of the cell, the first thing you see is basically the nucleus. Is basically the nucleus and the fluid portion, which is the cytoplasm. You understand? The most prominent structures, cytoplasm and the nucleus, is when you see the ultrastructure of the cell, and you can see the ultrastructure of the cell using the what we call the electron microscope. The electron microscope. It zooms to about 500,000 times the normal microscope, 500,000 times, okay? Now, that's the magnification of electron microscope. Now, what are the functions of this cytoplasm? The functions. Now, the first one is to maintain the shape and the consistency of the cell. Yes, it is there to maintain the shape and consistency of the cell. Another is for you to allow the chemical reactions necessary in metabolism. Now, you know that most chemical reactions that we perform take place inside the solvent water, right? They take place in the water. So that's why, since it contains about 90% water, then of course it's going to be the region where you know the chemical reactions in the cell will take place. Is that clear? Now, it is also the site for many metabolic pathways, including X glycolysis, protein synthesis, fatty acid synthesis, purine synthesis. That is, the cytoplasm will serve as the site for these things. Is that okay? And um, it is analogous analogous to the nucleoplasm and the content of the matrix of some membrane bound organelles, even though they have little differences. Is that clear? So but they are alike in functionality. The nucleoplasm, that is the fluid in the nucleus, and in the matrix of the mitochondria. What's happening there? So it helps to maintain the shape and consistency of the cell, allows for chemical reactions, and is the site for metabolic pathways. Now, the second layer that you must see is the cell wall. Okay. Now, the cell wall rather, the cell wall is only present in the plant cell. The cell wall is only available in the plant cell, not in the animal cell. Okay. Now, the plant cells are surrounded by a non-living and rigid coat called the cell wall, and that's one of the what differentiates cell wall from cell membrane or plasma membrane. The cell wall is dead. Is that okay? It's non-living, while the cell membrane is living. Exactly. So it's surrounded by a non-living rigid coat. Okay, so as you can see here, look at the structure. The ultrasound structure shows the middle lamella, the middle lamella, the primary cell wall and the secondary cell wall. Then you have the primary cell wall and secondary cell wall, and then the membrane. So this membrane, this yellowish and greenish part base here is the cell membrane. Okay, then from here upward, from on top of it upward, is what you call the cell wall. Okay, so now it is made up of proteins basically. Okay, proteins, cellulose, hemicellulose, pectin. Exactly. So those are the membranes that are the content of the cell membrane. Now, the cell wall of a differentiated cell has the following three parts. Number one is the medulla. So, and number two is the primary cell wall, and number three is the secondary cell wall. Now, in the middle lamella, we have the walls of contiguous cells are joined by the middle lamella. That is, cells that are placed side by side each other. They are joined by the middle lamella. Okay, that's made of contiguous cells beside each other. Now, it is composed of calcium and magnesium pectate. This is just a salt of pectin. Pectin is acidic, so pectin is the salt from the pectate. Pectate is acidic, exactly. So calcium and magnesium pectate um, are the components of the middle lamella. Then the primary cell wall is the one that lies between the middle lamella. It lies between the middle lamella and the secondary cell wall and the plasma lemma. Okay, so the plasma lemma is the cell membrane, like I said before. And now the primary cell wall is the one that is composed of the hemicellulose. 53% of the primary cell wall is hemicellulose, 30% is cellulose, 5% pectin, 5% protein, 7% lipids. It's very easy to cram. 53, 30, 5, 5, 7. 53, 30, 5, 5, 7. 53, 30, 5, 5, 7. 53 hemicellulose, 30 cellulose, 5 pectin, 5 protein, 7 lipid. Is that clear? All right. Then we have the secondary cell wall. It's the innermost layer of the wall. Now it is composed mainly of cellulose, but it's also contain lignin, suberin, and cortin. Now, this these lignin, suberin, and cutin are the things they are useful to us. They are useful products to us that we get from the plant cell, that we get from the plant generally, or a tree kind of, Those are products that we use, some of the products we use. So the functions of cell wall. Now the cell wall provides plants to a definite shape. Basically, it serves for support and rigidity. Is that okay? For the cell of for the wall of for the plant itself. So support, mechanical support in this way, is that okay? And strength to the tissues and organs of the plant. So, and what we mean by mechanical support is that it is what makes the plant itself strong and thick, you know. So that's one of the function primarily. Then, yeah, and that's the major function, just support, rigidity, strength for and of course, it also prevents desiccation. Yes, it also prevents desiccation. It also prevents desiccation. I didn't add that. Okay, then the plasmalemma, which is the cell membrane. The plasmalemma or plasma membrane. I told you can call it either of these three things: plasma membrane, plasmalemma, or cell membrane. Okay. Now it is the boundary of the cell, at least for the animal cell, that's the last portion you see outside. It is made up of a phospholipid bilayer. Don't forget that. Phospholipid bilayer. What does that mean? Phosphate group and lipid and fatty group. Bilayer, that is two layers. So that means that the plasmalemma is what? It has double membrane, right? It has double membrane. So we have seen the first guy that has double membrane, the first organelle that has double membrane in animals. So that is the plasmalemma. Plasmalemma. So now it has protein molecules scattered all over it. So it is studded. We say it is studded with fat and lipids and not lipids, carbohydrate. I'm sorry, studded with carbohydrate and protein. So we call them proteoglycans. Proteoglycans. They are special types of protein that are bound to carbohydrates um, that the plasmalemma is studded with. That is, it is, you know, those things are embedded in the plasmalemma. The composition is 40% protein and 60% lipid. So it's 40% protein and 60% lipid. 60% lipid because it is the lipid that constitutes the major part of it. We are going to see the diagram of that. You see it has that phospholipid bilayer is just talking about the fact that, so this is the way it looks like. Okay, so I'm drawing just two portions of the layer. So this portion here is going to be the phosphate group, and we call it the hydrophilic head. Hydrophilic. Excuse me. Let me use bro. Hydrophilic. Hydro means water, and philic from the word phileo means love, right? So water loving. So this is the phosphate region that loves water. This is another phosphate region that loves water. Then this portion here is the hydrophobic tails. The tail of this one is hydrophobic, that's why it's inside. The tail of this one is hydrophobic. Remember, the environment outside and inside the cell is going to be liquid, right? So because of that, these guys, only the phosphate group that is water loving will be in contact with that environment. But the hydrophobic tail, which is the lipid portion, remember the name phospholipid, okay? So it's the lipid portion that is water hating. So we say hydrophobic. Phobic means fear. Phobia means fear or hatred, okay? So that is um, it about the structure there. So we said the composition includes 40% protein and lipids 60%. So now The functions include the regulation of the movement of various molecules into and out of the cytoplasm. You know, there are two types of movements of substances within and without, into or out of the cell. And those are the active transport and the passive transport. Now, the active transport involves the movement of substances against a concentration gradient. Okay, so in this case, if they will be moving against a concentration gradient, then it requires energy to take those molecules from outside of the cell into the cell, or from inside the cell outside the cell. Is that okay? Generally, we say active transport is moving from lower to higher concentration. So energy is required and molecules need to drive this movement, okay? So this plasma membrane is studded with some molecules that can allow for active transport to take place, and of course also passive transport to take place. So the membrane, so we said the membrane is it also allows for passive transport, that is in form of diffusion and osmosis. Diffusion and osmosis. So movements due to concentration gradient, right? That is for both of them. Okay. So that is, it allows for movement in and outward movement of substances into and out of the cytoplasm. Then it also forms a protective barrier between the intracellular and extracellular environment. Yes, it forms a protective barrier. So it also does some protection work, especially in the animal cell where there is no cell wall. Okay. So now we have the structure of plasma lemma. Now you can see in this structure, first let's examine this basic structure down here. This is simpler. You're going to see the hydrophilic head and the hydrophobic tails, right? Hydrophilic head and hydrophobic tails. So these two together is what we call a bilayer, two layers, right? Then we had hydrophilic heads, hydrophobic tails. Okay, it's basically made up of the hydrophilic head, basically made up of phosphate, and the hydrophobic tail, fats or lipid molecules. Okay, now the carbohydrates. Now you can see here now that within them are studied these structures here, which are proteins. These ones here you are seeing here, they are proteins. So those proteins are now holding to themselves carbohydrates. That is, they are attached to carbohydrates. Exactly. They are attached to carbohydrates. Now also, cholesterol also build up within. Cholesterol also build up within the molecules here. So, but the point is, so these proteins are the ones that allow for active transport to be possible. Exactly. Then, of course, in between them, there are spaces in between the cell membrane. Of course, there are spaces in which passive transport naturally occurs. Okay, so carbohydrates, as attached to the protein. So so we call them proteoglycans, like I mentioned the other time. So we call these proteins that are studied within here, we call them integral proteins. That is, proteins that are attached within. So we call them integral proteins. All right. Then the proteins that are on the surface, we call them peripheral. When you say something is peripheral, we said, if I say we'll just do it peripheral, we just do the peripheral, it means we'll do the surface, on the surface level, we'll not go in depth. Exactly. So we call the one on the surface, the one like this one now on the surface, we call them the peripheral proteins. Exactly. Then you see here, if you look carefully, you're going to see these thread-like materials here. Thread-like materials, they are the cytoskeletons, and there are two. They are of two types. We have the microtubules and the microfilaments. The microtubules and the microfilaments. Now let's talk about the mitochondria. The mitochondria was first seen, so I hope you understand the cytoplasm, the cell membrane rather. Now let's see the mitochondria. Now the mitochondria was first seen by Kolliker in 1850. Colica was my friend in secondary school. So in 1850, so he first found them in muscles, and he called them sarcosomes. Okay. Now, Altman in 1990, 40 years later, observed this structure and named them bioblasts, while Bender in 8 years later now studied this structure and called them mitochondria. So it was Bender that renamed them mitochondria. So its name underwent different changes. We had the mitochondria, from mitochondria to sarcosomes. I'm sorry. First, sarcosome, from sarcosome to bioblast by Altman, and from bioblast to mitochondria by Bender in 1898. Okay, so it started with colica in 1850. Don't forget that. All right, then we have the structure. Mitochondria have a double membrane structure. Okay, it has an outer membrane and an and then intermembrane space, then an inner membrane. Exactly. That's double membrane too. Double membrane. You have outer membrane, intermembrane space, or perimitochondrial space, and inner membrane. Exactly. So the outer membrane primarily contains large number of integral proteins called porins. So the proteins that are found um, on the outer membrane of mitochondria are called what? Porins, and they allow materials in and out of the mitochondria. Now, the outer membrane encloses the entire organelle. It's the outer membrane that covers the entire organelle. So you're going to see something like some infoldings like that. So we call that the crystal. We're going to see that. Now the intermembrane space. So the intermembrane space, that is the space between this inner and the outer. This is the outer membrane here. Let me use different colors. So we have the outer membrane, this one here. Then the inner membrane is the one inside. Is that clear? Inner membrane. Then the intermembrane space, that is the space between inner and outer membrane. Okay, so this one, ions and sugars in the intermembrane space is the same as the cytosol. Yeah, it has it has the same materials, like I said before, as the cytoplasm. Okay, as we have in the cytoplasm. The cytoplasm is filled with the cytosol, so don't misplace it. The cytosol is just cytocell, sol. Sol is a type of colloid, do you understand, that exists. So we say cytosol, that is the fluid content of the cell, that's cytosol. We call it the cytoplasmic matrix. Yes, cytoplasmic matrix. All right, then we have the inner membrane rather, is the folded double lipid, phospholipid bilayer. You know, I told you um, the content of the plasma membrane are the same as the um, double membranes, double membrane organelles. Okay, then it is the site for production of ATP. If you don't remember anything about the mitochondria, remember that it is the one that creates the energy currency of the cell. The energy currency, that is the ATP, which stands for adenosine triphosphate. Adenosine triphosphate. Please, if you don't remember anything, just remember the primary functions I've been talking about. Adenosine triphosphate. Okay. Now, it also helps in the specific transport of proteins that regulate metabolites, metabolite passage in and out of the matrix. Yes. Some of the substances that are produced or produced by the some proteins are actually carried in and out of the mitochondria. Do you understand? Okay. So then we have the crystal. So the crystal is that inward folding that you see that, inside the mitochondria. So that is called the crystal. And matrix. The matrix is the space, the space that the fluid contains, that the fluid portion contains, that contains the fluid portion rather. So the crystal, this is the plural. No, this is the singular. Then the plural will be crystal. No, crystal is One is singular, the other is plural. I think this is singular, this should be plural. So cristae is the folding of the inner membrane that allows more space, more surface area, enhancing the ability to produce ATP. Yes, it enhances the ability to produce ATP. It enhances the ability to produce ATP. Okay? That folding, it increases. So we have um, the crystal, like I said, then the matrix is the space enclosed by the inner membrane. Yes, I already mentioned that is the space. Okay. Then the chemical composition of the mitochondria, we have protein 70%, lipid 25 to 30%, RNA 1%, and DNA less than 1%. So you see, it has its own genetic material. Now, there's something about the mitochondria. The mitochondria is the mitochondria that are in your body. All the mitochondria that are in your body are from your mother, and the reason is because during the fertilization of egg and sperm, the egg, the only thing that the sperm contributes is the genetic material of the father, but the egg is the one that contributes. It contributes more percentage, a greater percentage. I've actually forgotten. I think is the one that contributes about 70, 60, 70 to 70%, because organelles like mitochondria are present in the egg cell, and the genetic material inside the nucleus, of course. Okay, so but the point is this: that if the mitochondria of the mother has genetic disorder, then there is a high likelihood of transmission of that genetic disorder. So note that that's why we call it maternal inheritance. Maternal inheritance. That's why we can trace the genealogy of people by using their mitochondrial DNA. Do you understand? Because it will show us the maternal the genealogy. Okay, so it has its own genetic material, DNA and RNA. We call them mRNA, dRNA. Now people usually confuse. So when I say mRNA, for example, now we have mRNA. There is one called the messenger RNA, and there's one called mtRNA. That's what we call it. Yes, some textbooks use mtRNA. That is mitochondrial RNA. Okay, so don't confuse them. M is messenger RNA, and MT mitochondrial RNA, mtDNA, that's mitochondrial DNA. All right, the functions include so I said the important and main function is to produce ATP. So it is called the powerhouse of the cell. Don't forget. It is called what? The powerhouse of the cell. The mitochondria is called what? Powerhouse of the cell. Then the Krebs cycle of cellular respiration takes place in the mitochondrial matrix. Yes, you remember in cellular respiration, we have glycolysis. Then the glycolysis now moves to the product of glycolysis of the glycolytic pathway now moves to the Krebs cycle. So glycolytic pathway takes place in the cytoplasm, while the Krebs cycle is the one that takes place in the mitochondria, because it's the Krebs cycle that actually produces about 36 ATP, more ATP than glycolytic pathway produces. Glycolytic pathway ends up with a total ATP, a net ATP of 2 ATP. While Krebs cycle that happens in the mitochondria ends up with the 36 ATP. So don't forget that the Krebs cycle takes place in the mitochondria. Then it regulates cellular metabolism. Yes, it also regulates cellular metabolism. Okay. Then it plays important role in cytoplasmic inheritance. It plays important role in what? Cytoplasmic inheritance. Then it is the site of metabolic pathways, okay, such as TCA, that's Try, what is TCA again? We have the TCA cycle, urea cycle, FA oxidation, that's fatty acid oxidation, um, urea cycle, the TCA, TCA, that's, yeah, that will be assignment, just find out what the TCA cycle is. I already covered it. I covered some metabolic pathways in the biochemistry series. What is TCA again? Why am I forgetting? Okay. So the Krebs cycle is also called citric acid cycle. All right, now the structure, look at it here. So in the structure, you're going to see the outer membrane. You see the outer membrane, this is the outer covering. Then we have the inner membrane, this is the inner membrane here, the bluish portion here, that's the inner membrane. The in between, the space in between, that'll be intermembrane space. Then the space that is filled by this liquid is called the matrix. Do you understand? And then we have ribosomes around it, the mitochondrial ribosome, and then the folding here you see is called the cristae. All right. Then then there are particles here that we call the ATP synthase particles. That is, ATP synthase help in the synthesis of ATP. Exactly. So crystal is singular. Yes, cristae is plural. So that's correct. I said that. All right. Then we have the intermembrane space and all of that. So that's the structure of the mitochondria, the ultrastructure of the mitochondria. Now let's go to the plastid. We have the chloroplast. Now, I didn't cover it here, but I think I covered in one of our video where I talked about the leucoplast and the ameloplast, okay? Because this is not the only plastid that we have in the animal, in the plant cell actually. Now, our animal cells don't contain chloroplast. So, you know, we have seen two things that are not in animal cells now. We have the cell wall and the chloroplast. So far, those two things are not in the animal cell. Okay, so now it is found only in plant cell and plant-like protists, like the blue-green algae, the no, even though in the blue-green algae we don't call it, it's not an eukaryote, so it doesn't have blue-green algae does not have chloroplast. Remember, chloroplast is membrane-bound, so it only have structures like thylakoid-like structures. You remember that the if you okay. First, let me show you this now. Inside the chloroplast are thylakoids. And in the thylakoid membrane, we have chlorophylls studded. It is studded with chlorophyll. And it is this chlorophyll that is a green pigment that makes the plants green, exactly. So inside the cell of, in the cell of prokaryotes, they only have thylakoid-like structures. They don't have chloroplast, so don't mix that up. I was talking about protists like the chlamydomonas, so that's the true protist. And it's flagellated, so it's a flagellate. The other true protist that will have chloroplast, that is Iglina. And of course, the rhodophytes, the chlorophytes, Spirogyra, for example, the phaeophytes, all those guys, do you understand? They are the algae that will the algae that will contain chloroplast. So it contains chlorophyll, hence it imparts the characteristic green color to, to plants and carry out photosynthesis, the ultimate source of all organic compounds. Yes, so it is photosynthesis that brings about all organic substances that we I mean, that is organic substances in plants. Is that clear? Okay, so and of course, through primary production is the process by which all this, the first stage of production, do you understand? Before energy is now being transferred and converted into different forms in us, I mean, when we eat those plants. That's what I mean by ultimate source here. Is that clear? But the point now is that, so the word chloro actually means green. So if you remember chlorine, the color of chlorine, that's greenish yellow, right? Then plast means plastid. Plastid means something that is housing something. That's made of plastid, that is covering or embedding something. Now we have the average cell may have 20 to 40 chloroplasts. Yes, the average plant cell may have about 20 to 40 chloroplasts. And the chlamydomonas have only one chloroplast, for example, because it's a protist. Now, what are the contents? The contents include protein, lipid, chlorophyll 5 to 8%, carotenoids 0.7 to 1.1%. It also has its own RNA and DNA. Okay, now let me quickly take some time to, I will explain this in the physical class later, but I just want to take some time to brief you through the what is called the endosymbiotic theory. So according to the endosymbiotic theory, for example, endosymbiotic theory, let me write that, endosymbiotic theory. I only explain this one. I will explain MIT, membrane invagination theory, in the physical class. Now, endo means inside. Symbiosis means an association where both of them are mutually benefiting, right? Okay. Now, the theory actually talks about how the first cell, the first prokaryotic cell, how it became the first eukaryotic plant cell. Is that okay? That's what the endosymbiotic theory says. And it says that the first prokaryotic cell, or it explains that the first prokaryotic cell was actually, um, you know, more like it was independent. It wasn't efficiently productive in the area of food production. So so it now engaged in a symbiotic association with the chloroplast, and you know that the chloroplast can produce food, right, through photosynthesis. So according to that theory, the chloroplast was existing on its own, like as a cell initially. So it was when they needed to, you know, but it wasn't also efficient in the area of its protection. So the first prokaryotic cell and chloroplast merged together, and with other, as well as other organelles such as mitochondria. These organelles, according to that theory, were existing on their own before. So because of what the different things that they need and the different purposes that they can serve to each other, then they engage in symbiotic association, so that, for example, the chloroplast will be producing food for the prokaryotes, and the prokaryotes will be serving as, will be protecting the mitochondria and the mitochondria and the chloroplast. So the mitochondria too will be providing what? Energy, isn't it? So and that theory is quite true, because you see that both structures that I've talked about so far, mitochondria and chloroplast, you see that they have their own genetic material. They have their own genetic material, they have their own plasma membrane and all of that. So that is what shows you, that is one of the proof for the endosymbiotic theory. So we see the origin and evolution, that is plastid evolution. So it begins with the cyanobacterium. So a cyanobacterium now developed into the chloroplast. Is that okay? So that theory that I just mentioned, actually, this is the origin. Do you understand now? How the chloroplast was first formed. Do you understand? So the cyanobacterium develops into this. Then um, the chloroplast will perform function of photosynthesis to produce starch, to produce lipids, to very, very minute degrees. Okay, the chloroplast is majorly for proteins, for starch metabolism, production, food, glucose. Is that okay? Then pigment synthesis, genetic system, and etc. So all these guys will now be stored. The chloroplast, depending on what it produces, is that okay? It will now be it will now specialize. You understand? To undergo specialization. So we have plastids that now form chromoplasts. Chromoplasts. So chromoplasts are structures that allow so for example, carrot is orange in color because of carotene that is stored in its chromoplast. The word chromo means color, colored. So if you hear chromosome, that's colored body, color. So we said color, chromoplast, they store colors that help in pigmenting fruits and the products of the plant. Then, so it means that those plant structures are going to be rich in chromoplast. Then we have the amyloplast that store starch. You understand? Amylo, from the word amyl or amylase. If you remember amylase, the enzyme that degrades um, carbohydrate. So amyloplast. We have the etioplast. That is the light-dependent development. Do you understand? For for reception of light. Then there is the proplastid. So that's development. That is the one that will form into a plastid again. Do you understand? Okay, so that's primarily it about the chloroplast. The stroke is so now the structure. The structure. Now we have it has a planoconvex lens-like structure. Planoconvex means this is biconvex. Yes, biconvex. This is biconcave. Exactly. Now planoconvex means it has a plane from the word plano, that is plane, and convex. Plano concave will mean a plane and a concave. Is it clear? Plano convex. Then biconvex will be like this. Biconvex, like a lens. Okay, so plano convex like structures, because if you look at it, you see that a side is rounded, the other side is flat, like that. Do you understand? It has double layered envelope, like we said, so it has a bilayer also. Now look at this. You are going to see outer membrane, intermembrane space, and inner membrane, just like the mitochondria. Then the space here, it is in this thing, it is in the mitochondria, it is called the matrix, but here it is called the stroma, the fluid filled portion. The fluid filled portion is that okay, it's called the stroma. Then you now see these disc-like structures or planoconvex lens-like structures. They are they are called the thylakoid that I was talking about, thylakoid. Now, one of them is thylakoid. They usually stack up themselves. They stack on top of each other to form the granum. They form the granum, the plural of which is grana. So we have them here, grana. These are grana, is that clear? Now, then the space in between them, now when you break the the thylakoid, you break it open. The space in between the top and bottom layer is called the thylakoid lumen. It's called the thylakoid lumen, okay? Then of course, there is this one that connects them. We call it the stroma thylakoids. The stroma thylakoids are the one that connects each of the stack of thylakoids. Okay. So we said it is double layered, that's double layered envelope, granular stroma or matrix, then lamellar system, and that is the lamellar system inside the structure, osmophilic globules, plastoglobuli, and chloroplast DNA. These are the structures that you find inside. Then the function, the main function, remember, the main function, photosynthesis, food production for the plant. But mitochondria, main function, ATP synthesis. It contributes to heredity in form of cytoplasmic inheritance. Yes. Okay, in fact, I'm going to be discussing the cytoplasmic inheritance. So here too, cytoplasmic inheritance is just like the mitochondrial inheritance. Only the female rather, contributes to the cytoplasmic inheritance. Just like the female, only the female contributes to the mitochondrial. Do you understand? Mitochondrial genes. Is that okay? The inheritance of the mitochondrial genes. Only the female. Only the female. All right. So let's quickly see that. Okay, no, no. Here is the energy transduction system. This shows how the chlorophyll works to produce produce food. This is just the process of photosynthesis, exactly. So you see the photon of light is going to hit, is going to strike the photosystem 2. Photosystem 2 first, because they were named in the order that they were found, that they were discovered. The photosystem 2 is called plastoquinone oxoreductase. But that's just mouthful. You don't need to cram that. But just so you know that it has a special name, plastoquinone oxoreductase. Okay, now I now said that, okay, so it's going to energize molecules. The photosystem is going to energize electron carriers, and those will lead to the oxidation, the reduction of water, and then leading to production of protons, and then the energy from those ions, from those electrons, will be transferred and transferred, you know, and until it gets to go through the cytochrome complex, and then move to the photosystem 1. Is that okay? And then the photosystem 1 will lead um, to the what's it called, fixation of ribisco. What does that stand for again? Ribisco, that's ribulose. Why am I forgetting these names? I've covered these two in photosynthesis. I'm going to post the video soon. Rubisco, that's just it. Is a molecule, a moiety that helps, is a 5 carbon chain molecule that is used in the production of glucose. Is that clear? Rubisco, that is ribulose carboxylase, right? Yes, ribulose carboxylase, yes, something around that. Okay, so by that, by the use of the reduction of, it will also aid the reduction of NAD plus to NADPH. That is nicotinamide adenine dinucleotide phosphate hydrogenase, when it is reduced. Then the ATP synthase complex will be the one driving this process, because the production of food here is, requires energy. So you see that it uses up energy. You understand? Okay. So this is the machinery that drives the whole process, basically. Okay. All right. This shows the complexes distributed in the thylakoid membrane. The complexes. Okay. Let's see the material inheritance of chloroplast genome, the material inheritance. Now, nuclear genomes of higher eukaryotic organisms are diploid, and they show sexual reproduction. Okay, yes, they show sexual reproduction. Then, gametes result from meiosis. Do you understand? Gametes that result from meiosis are haploid. I told you that the word haploid means from the word haplus, it means single, and by ploid we mean chromosome number. So single set of chromosome, that is haploid, and they are product of meiotic division. Then we have fertilized zygotes inherit one nuclear allele of each gene from each parent. But fertilized zygote will inherit one number of allele of each gene from each of the parents. But cytoplasmic organelles, such as multiple homogeneous genomes, are inherited only from female parents. That's cytoplasmic inheritance. So you see that nuclear material inheritance is different from the cytoplasmic inheritance. In cytoplasmic inheritance, it deals with how that only the female, I've mentioned this before, only the female pass to their progeny the substance contained in their mitochondria and cytoplasm. Okay, so for example, in tobacco and many other plants, mitochondria and chloroplast genome are specifically degraded in male gametes. Yes, they are broken down so that only the genetic material is available for transfer. But in the female, it is still available before fertilization. So once fertilization occurs, the progeny is still going to retain the material from the mother. And of course, there are many exceptions. So now there are many plants that do not, follow that pattern. Now, this shows the allele, how genetic material is transferred. So this shows the male parent, and this shows the female parent. So you see here that before fertilization, we have the ABC of the cell, of the nucleus. This is the nucleus here now. And then you are going to see the chloroplast and mitochondria. For the male, you see it's not transferred at all. But those of the female, we have the nuclear material transferred, those of the mitochondria transferred, chloroplast also transferred. You understand now? So the diploid zygote will now have AA, BB, DD of a diploid nucleus and CP, empties of the cytoplasmic genomes. Is that clear? Okay, so that is the way it's being transferred. Only the female will transfer its genome. So this place is also explaining the same thing. Now let's take a look at the endoplasmic reticulum. Now the endoplasmic reticulum is a web-like series of membranes within the cytoplasm in the form of flattened sheets, sacs, tubes, and it creates many membrane enclosed spaces. It spreads throughout the cytoplasm and has connection with the outer membrane of the nucleus and the plasma membrane. Okay, so in fact, the word ER means endoplasmic reticulum. Endo means inside, cytoplasmic. That is, we remove the cyto from the plasmic. I'm talking about the etymology of the word now. Endoplasmic reticulum. Reticulum simply means network. Reticulum means connected, connections. So network of membranes inside the cytoplasm. That's the meaning. Network of membranes inside the cytoplasm. Now, it refers to interconnected network of tubules and vesicles, that is cisternae. Tubules and vesicles are connected together. Is that okay? So usually, according to the MIT, the membrane invagination theory, they are derived from the the nuclear envelope. Thank you. From the nuclear envelope. Okay, so it's a network of interconnected tubules and vesicles. Okay, so as you can see here, now this place typically shows the MIT. Okay, so according to the MIT, the nucleus began by the plasma membrane enclosing the nucleus. Okay, and after enclosing it, it now moves to the center of the cell, and of course, other materials are now synthesized. So it now has holes around it. That is the holes such as you can see here, spaces that we call nuclear pores. Exactly. So that nuclear pore is so that substances that are produced within the nucleus, they will be able to move out of the cell. By the time they move out of the cell, and they'll be able to move out of the nucleus rather. So after a long while, this or after a while, I'm trying to explain the MIT now. The nuclear pores, the nuclear envelope, this guy, nuclear membrane or nuclear envelope, will now um, will now lose its new nuclear envelope would have been secreted and it would have covered it. So the old one will now form around the nuclear envelope, which is what will now become the endoplasmic reticulum. This time it will be the rough endoplasmic reticulum because inside the nucleus is the nucleolus, where the ribosome is secreted. So ribosome will flow out of the cell and will be formed on the endoplasmic reticulum. So we'll call it the rough endoplasmic reticulum. But also, there are some of these endoplasmic reticulum that will not carry ribosome at all. So those ones will be the smooth endoplasmic reticulum. So their functionalities will be different. Then you will now see that the structure of smooth and rough, they are very similar, and they are very similar to the structure of the Golgi apparatus, right? So according to the MIT also, it is now the smooth endoplasmic reticulum or the endoplasmic reticulum that does not have ribosome that will eventually become the Golgi apparatus. Remember, the Golgi apparatus was named after Camilo Golgi. So, and these guys will now form vesicles because it processes materials in the cis trans direction. So it will form vesicles. Those vesicles will now transport substances in and out of the cell. Some of them become lysosomes that break down substances. Exactly, that detoxifies. Some of them become peroxisomes and all of that. Okay, so you can see the MIT theory, the endo membrane theory also makes sense. So there are two types, as I said. We have the rough ER that is studied with ribosome, and the rough ER is the site of synthesis of many proteins. Yes, it is the site for synthesis of many proteins because the ribosome is the one that creates proteins. So if the ribosome creates protein and the endoplasmic reticulum is studded with protein, with ribosome, then it makes sense to say that endoplasmic reticulum is also important in the formation of proteins. So all ribosomes on the rough ER are actively involved in protein synthesis. Then you have the smooth ER. It is the site for synthesis of steroids and other lipids. Calcium ion storage in muscles here also, yes. It also helps in the storage of calcium ion, that is the smooth ER. So the two major functions are rough ER, synthesis of protein, smooth ER, synthesis of steroids and storage of calcium. Then smooth ER also helps in detoxification. That's why it is most abundant in the liver. Okay, I didn't even mention all those things. Mitochondria, for example, will be most abundant in very active sites, very active organs, such as the mitochondria. That will be heart, in the heart region, of course, also in the liver. Mitochondria will be very much there. Also in the kidney also, but it will be primarily more in the heart and in the liver. Then the endoplasmic reticulum, for example, will be much in the liver, will be much in the liver, will also be in the kidney, yes. It will also be in the kidney. Like it will be much. It's in all cells, exactly. I'm talking about their degree of abundance now. Okay? Now we have the functions. Rough ER, synthesis of protein. Smooth ERs, synthesis of phospholipids, cholesterol in many tissue, and steroid hormones, such as the adrenals and gonads. Then the smooth ER, it is the site for glycogen metabolism. It is the site for glycogen metabolism. So if you remember the pathway of fats from fats to glucose that we call gluconeogenesis. No, glycogenolysis. There's gluconeogenesis and there's glycogenolysis. Okay, that's a breakdown of glycogen or glycogenolysis. Yes, breakdown of glycogen, then gluconeogenesis, production of glucose from new origin. So that the smooth endoplasmic reticulum it has a role to play in these metabolic pathways. Then we have it also removes the phosphate group from G6P, that is the glucose 6 phosphate. That's glucose 6 phosphate. It helps in the removal of phosphate group in order to release free glucose in the blood. Is that clear? So free glucose in the blood. Right. Is that clear? Now, in liver and kidney, it detoxifies drugs. Yes, in the liver and kidneys. I said it's also abundant in liver and kidney. Yeah? So it detoxifies drugs, toxins, and carcinogens. Carcinogens are things that are liable to cause cancer in the cell. Then it also helps in the circulation and transport of substances. It also helps in storage of protein and minerals and a large surface area for enzyme action. Yes, it also assists in, provides a large surface area for enzyme action. Now we have the Golgi complex. Now consists of 3 to 20 cysteinae. So remember, the infolding of the mitochondria, cristae, or crista for singular, while for ER and Golgi apparatus or Golgi complex is the cisterna. Is that okay? Crista for mitochondria, cisterna for ER and Golgi apparatus because they have the same origin. So it consists of 3 to 20 cisternae, small flattened and membranous sacs, prominent in cells that secrete proteins. Yes, it is prominent in cells that secrete proteins and accept vesicles on one side and produce vesicles on the other side. So we call it the cis direction. That is where it accepts vesicles. And to process the vesicle through the other direction, which is called the trans direction, like we saw in the slide I showed us the other time. Okay, we can also see it here. So this is the secretive vesicle that it takes from this side, then it's releasing it in the trans side. Okay, so it takes in the cis, release in the trans, or takes in the trans, release in the cis. Is that clear? So that's that. Transport of vesicles from the Golgi. We have new vesicle forming. So you can see that it forms vesicles too. These vesicles are, according to the MIT theory, is what later becomes the lysosome, the peroxisome, and all those vesicles around the body. Now the functions. What are the functions? We have the modification of proteins and lipids. Smooth endoplasmic reticulum produce lipids and steroids, right? While the rough produce proteins, right? So these ones will now be modified. That's why it's processed. That's why we call it processing. It will take those proteins and lipids from the endoplasmic reticulum in the cis direction and go and process it in the trans direction for storage and packaging of molecules. Is that okay? Is that okay? So we call it the post office of the cell. Yes, the post office of the cell or the cell messenger, according to some textbooks. Okay, so we have ribosomes. Now the ribosomes help in the protein synthesis. It's the one that produces proteins. It produces proteins. Then we have two subunits, the large and the small, each made of protein and ribosomal RNA. Yes, there are two units, subunits. We have the large and the small units. Yes, it's just their size that differentiate them. And they are made up of protein and ribosomal RNA. Ribosomal RNA is the precursor for all amino acids, that directs the formation of the amino acids. Just check my video on transcription and translation. You will understand what I'm talking about. Now, it is granular structure. It is the granular structure present on surface of ER and also as free. Yes, it is free in the it is free in the cytoplasm. It was first observed by George Palade in 1953. Now, non-membrane bound organelle. Yes, it is non-membrane bound. There are three non-membrane bound organelles. We have not just three, there are many, I think there are about 4 or 5 non-membrane bound, but the three I usually remember are the ribosome, centriole, and the centriole, and cytoplasm, yeah? Because the vesicles, peroxisome, all those guys are single membrane bound, have single membrane, then the other ones, mitochondria and nucleus and chloroplast, have double membrane. Okay? So the size is about 22, approximately 22 by 32 nanometers. So that's very small. Contains 85% RNA of the cell. So most of the RNA of the cell are found in the ribosome. So, and I've told you RNA is the precursor to mitochondria, amino acid rather, amino acid, which is um, the end product, the simplistic version of of proteins. Okay, so that all those all add up. Then the functions, subunits associate when they are synthesizing proteins. The subunits, they associate when they are synthesizing proteins. And protein synthesis occur on ribosomes that are free floating in the cytoplasm and on ribosomes attached to the ER. The subunits associate when they are synthesizing proteins. Don't forget that. So, and then I've covered that in the transcription and translation video. Now, the protein synthesis occur in ribosome that are free floating in the cytoplasm and on ribosome that are attached to the ER. So those two they produce protein. Then rRNA is synthesized in the nucleolus. So the rRNA is the one that works. That I've told you all this. That is the precursor to amino acid, and then that it directs the type of protein that will be produced. The rRNA, that is, so it is produced within the nucleolus, that is rRNA. Now, so this is the ribosome, basically. So you see, this is the ribosome. We have the small unit, the small subunit, and the large subunit. The small subunit and the large subunit. Okay? So you can see there's a signal sequence, that is the sequence that comes from the nucleolus. Okay? The sequence that comes from this nucleolus, you're going to see that it will direct the production of this thing. This green structure you're seeing here is just a polypeptide. A polypeptide is just a peptide is amino acids that are attached together. Amino acid, amino acid, amino acid. You understand? Amino acids. So, but the difference is that these amino acids are no longer in their deuterionic form. They have now been packaged. Do you understand? There are 4 stages of the packaging of protein. So of amino acid into protein. So these ones are already in their beta pleated sheets. They have formed what is called the beta pleated sheets. So those beta pleated sheets are the ones, so that entire sequence together to form a larger, longer molecule is what we call polypeptide. So you see that is the machinery. It is entering, you see that this thing is entering into it and it is producing this long chain. You understand? So this is the signal molecule, that directs what it should produce. Okay, this is actually the membrane of the endoplasmic reticulum. And this the yellow part is the cytoplasm. Now let's talk about the lysosome. The lysosome refer to vesicles filled with digestive enzymes. Yes, they are primarily filled with digestive enzymes. In fact, the word lysosome, lyso means splitting, splitting. Is that okay? Then soma refers to the word body. Okay, so splitting bodies because they are made up of lytic enzymes, enzymes that help in splitting or breakdown or digestion of substances. So they are used in intracellular digestion, that is within the cell digestion, and particles can be taken into cell by phagocytosis and vesicle fused with lysosome. That is, that is, if for example now, a substance is supposed to be detoxified, is to be detoxified. So the Golgi, the smooth endoplasmic reticulum might package that thing in vesicle, or the Golgi apparatus do the same and transfer that vesicle. That vesicle will now move to the lysosome. So what the lysosome will now do is that, so if this is the vesicle that contains substances to be detoxified, to be digested or detoxified, the lysosome too contains lytic enzymes. So the two of them will now come together. By the time they come together, they are going to form something like this. So this guy will now embed, empty, will swallow up this guy. Usually, in fact, the vesicle is smaller. So what the way it forms is even like this. It's mostly like this. It phagocytizes. The word phago means eat, to eat. So phagocytosis. Let me first explain this. So this is the lysosome now. This lysosome will now form around the vesicle, to form around like a mouth around the vesicle. I'm coming. Then the vesicle that contains the material is here. So it's going to now fuse with its membrane and invaginate and swallow it up. That's how to think of it, swallow it up. I told you the word phago means to swallow or eat. Cyto, that is cell. So cell eating, that's the process by which lysosomes, digest these vesicles and detoxify and break their contents down. So if that is the case, therefore, which of the cells will contain more of this lysosome? God bless you. That makes sense. White blood cells. Do you understand? The white blood cells are going to be abundant in them. The liver are going to, the cells of the liver, because they help a lot in detoxification, they are going to be highly abundant in what? Lysosome. Now, the components of the organelles can be recycled after digestion by lysosomes. And the functions involve garbage disposal, breakdown of bacteria entering cells, worn out organelles, intracellular digestion. So the function is as follows. You see the rough ER as processed substances and given to the Golgi apparatus. Now the Golgi apparatus will now package this in vesicles, okay? To package this in vesicles. So if such substance is to be removed from the body, now it could, you know, it could be that the material that is inside, for example, lysosome also function in autolysis. I didn't mention that. Now, autolysis means self. Auto means self. Lysis, splitting. That is, it destroys the cell itself. So, worn out cells are destroyed by the lysosome themselves. They will just release their digestive enzymes inside the cell, and the cell will divide and you know die off. So, autophagy is self-eating. That's the meaning of autophagy. That is, the lysosome will engulf damaged organelles within the cell. So you can see here that there are organelles here. There's an organelle here that has probably damaged. So it is packaged in a vesicle by the Golgi apparatus, and it undergoes digestion. So then it becomes a vacuole, as you can see, or it just goes from lysosome. The lysosome will take up these substances, and if it needs to be, to be digested, basically that's the point, to be digested and broken down. Do you understand? To be broken down. Okay, yes. So the food also comes in through here. Then after that is also phagocytosis, or by phagocytosis. That is the cell, the membrane of the cell will also invaginate a little, take up the food, form membrane around it, and then this place will disappear. Then this thing will fuse inside and go inside and be packaged as a vesicle. Do you understand? It will be packaged as a vesicle. Do you get it? So as it's being packaged as a vesicle, the lysosome will also take it up and digest it. Do you understand? That's the sequence. Right now we have the vacuole. The vacuole is large in plant cell, very small in animal cell. It helps in the osmotic regulation of the cell. It's also a membrane-bound sac. It contains liquids and solids and larger than vesicles. Yes, it's larger than vesicles. The function is storage container for water, food, enzymes, waste, and pigments, etc. Actually, it regulates osmotic condition, regulates the osmotic condition of the cell. Then we have the centriole. Now, it's part of the specialized region of the cell called the centrosome. It's called the cell center, yes. It's usually located close to the nucleus at the center. Do you understand? So we call it the centrosome. Central means center, some, body, that is the body in the center. Now it is found in animals and most protists, but not found in the plants. Is that clear? Centrioles are involved in production. Centrioles are non-membrane bound. Production of microtubules, yes. They are involved in the production of microtubules. Okay. Microtubules are made up of actin fibers. Microtubules have many functions, including moving chromosomes during cell division. Central structure, 9 triplets of microtubules, yes, surrounding a hollow core. In fact, they have a particular dimension that they follow in the arrangement of that 9 triplets of microtubules. That is 9 sticks. They look like sticks. You understand? So the arrangement is such that both of the centrioles are at 90 degrees to each other. They are at right angle to each other. Okay? So it is similar to the basal body of flagella. That is the microtubules, which make up the centriole. Okay, so during cell division, they play an important role. Is that okay? The structure is like, okay, so you can see this. You can see this. So they are in triplicates: 1, 2, 3, 4, 5, 6, 7, 8, 9. You understand? They are in triplicates, 9 triplets. That's what I called it. So centrioles are short cylinders arranged at right angles to each other. I already said that, at right angles to each other. Now the cytoskeleton, a network of protein filaments that extends throughout the cytoplasm, anchored to plasma membrane and dynamic structure. The cytoskeleton are of 3 types. We have the microtubules, intermediate filament, and microfilaments. Their sizes decrease as you go down. We have microtubules, they are larger, or would I say fatter? They are not fat, but relative to the three. This has the larger size. This one have tiniest size, and this has the tiniest size. Okay, they are probably made up of almost the same thing. Now, of the same thing. Now, the microtubules are long, hollow, unbranched polar cylinders made up of protein called tubulin. That's what we call the microtubules. Microtubules. They are made up of tubulin. And there are 2 types of tubulin. We have the alpha and the beta tubulin. It is the largest of the cytoskeletal components. Cell skeleton. Is that okay? Components. Major components of axons and dendrites. Yes, the microtubules are the major components that form the axons, in this that attaches the cell body, that attaches to the cell body and connects one nerve to another. They connect one nerve to another. So it is it forms something like tentacles, right, around the cell body. So that's this body here, this axon here is made up of microtubules. That's the major component. Then microtubules in function help in structural support and maintain the cell shape. Yes, That's their major function, maintenance of the cell shape. And it helps in movement of organelles, secretory vesicles, and exocytosis. Yes, the process is simple. It's just like when something is floating. So as it's floating, to be hitting the microtubules. As it's hitting the microtubule, it will be bouncing away and moving towards where it's supposed to move. So we have formation and function of mitotic spindle. Yes, during cell division, it gathers mitotic spindles, to form, these spindles that form during cell division, okay? So that it will attach to the chromosomes, you understand, at the kinetochores. So this organization of these things is done by the microtubules, um, by the centriole. So these microtubules are those things you are seeing there. Those spindle fibers are made of microtubules. Then we have movement of cilia and flagella. Then we have intermediate filaments. They are polymers of long rod-like proteins. You see, they are still proteins. These proteins are made up of keratin, desmin, and neurofilaments. And these filaments are thicker than microfilaments, but thinner than microtubules. Is that clear? They are thinner than microtubules, like I said. That order that I gave you, microtubule, intermediate filament, and microfilament, that's the order of their decrease in size. Function, also for mechanical support and shape, helps in intercellular attachment, and provides strength and rigidity to neurons, and major structural role in skin and hair cells. The microfilaments, they are the thinnest elements in the cytoskeleton that consist of actin proteins, and they're made of beta and gamma actin. Remember, microtubules, they are made up of alpha and beta tubulin. These ones are made up of beta and gamma actin. Then they form a meshwork under plasma membrane. Then they are stress fibers. Okay? Then that was what we saw under the plasma membrane, if you remember. Okay? Their function includes mechanical support for basic strength and shape of cell. Um, X microvilli is rich in microfilaments, then involved in muscle contraction, cell division, and cell locomotion. So these are the structures. We have the microtubules here. You can see the microtubules, intermediate filament, you see them, and the microfilament, you see. So look at their sizes, they differ. The sizes differ. And you see that microtubules are closer to the center than the intermediate filament, and the microfilaments are the ones that are closest to the cell membrane. So microtubules, see their sizes again. Okay. These are the actin filaments, these are the intermediate filaments. All right. My God. Okay, we still have a lot to do. So, what are the functions? They provide shape to cell and act as internal framework, helping uptake of materials into the cell, and helps in internal movement of cell organelles, movement of cells and muscle contraction, and helps in cell division. Then the peroxisome. Okay, peroxisome are actually they are known as microbodies in the cell. They are subcellular respiratory organelles, and they have 0.5 micrometer in diameter. Now, they are predominantly present in hepatocytes and tubular epithelial cells. Okay? Hepatocytes, liver, right? Liver cells, so they are abundant in the liver and the kidney. Peroxisome. Why? It's because their major role is what? In detoxification. In detoxification. They use peroxidase or catalase in detoxification of H2O, H2O2, that's hydrogen peroxide, to form H2O and O2 and oxygen. Is it clear? Let me rewrite that. H2O2 to form H2O plus O2. So or the enzyme, there's a catalyst here, that's a catalyst. Another enzyme that does the function is peroxidase. So they create, so they are oxidases, exactly. They contain several oxidases. So they create H2O2 as a byproduct and degrade it with enzyme catalyst. So the function, oxidation of amino acids, oxidation of long chain fatty acids. Yes, they help in the degradation of fatty acids. So remember, so they will break down fatty acids, but they don't use ATP. So know that they don't release ATP, they don't produce ATP rather. Because the final process of this degradation, they only break very long chain fatty acid into medium chains so that those medium chain will now go to the mitochondria for breakdown to release, to form glucose precursor, that is G3P. G3P, that's, how am I forgetting this? Glycerate 3 phosphate. The analogous of dihydroxy acetone phosphate. Anyways, the point is they don't use ATP because they don't break down this thing fully to release energy. They will just break it down into chunks, and those chunks will now move to the mitochondria for the release of energy. Okay? So they protect cell from toxic effect of H2O2, which are they are byproducts of some metabolic pathways. They are used for removing reactive compounds from the cytoplasm. Yes, they are used for removing reactive compounds from cytoplasm. They essentially contain two types of enzymes, which are oxidases, such as the peroxidase and catalase, which act on hydrogen peroxide to liberate oxygen. We have the spherosome. Spherosome is just according to the way the shape is spherical, that is sphere, and zome, that is body. So they are vesicles surrounded by a membrane found in plant cells. They are found only in plant cells, and their composition include 98% lipids plus some acid phosphates. And their function is just the storage of lipid in the plant cell. Then we have the nucleus, is the control center of the cell, and there has the information needed to oversee the chemical reactions in cells. It is prominent, that is big, and characteristic features. It has, that is when you see the cell under under a simple microscope, what you will see is just the cytoplasm and the micro and the nucleus. That's what I explained in the beginning. So eukaryon means true nucleus, and it is very essence of the eukaryote, the membrane-bound nucleus. The function includes storage of genetic information, control cell division, directs functioning of cell, that is, provides template for RNA and protein synthesis. We call it the central dogma of molecular biology, from DNA to RNA to protein. Okay, then they physically separate DNA from cytoplasms and complex metabolic machinery. And nuclear membranes serve as boundary. So as you can see here, you see the nuclear envelope that has pores, for outlet of substances. Then nucleoplasm, which is the fluid interior portion. Nucleus, the dense cluster of RNA and protein would have, for production of ribosome. Then we have the chromatin and DNA plus proteins. So you see the structures, they are embedded inside the nucleus. The nuclear envelope, that's the inner and outer nuclear envelope with perinuclear space. Basically, you can just read on this. They are just describing the nuclear envelope. We have the nuclear pore. Is the most distinctive feature of the nuclear envelope. And that's because it is holes. It's a small cylindrical channel and has direct contact between the cytosol and the nucleoplasm. It is readily visible and freeze under freeze fracture microscopy. And the density, you know, it depends on the cell type and the activity of the cell. So the nucleolus is the one that produces, we call the ribosome factory. It's a large and prominent structure, doesn't have membrane because it's inside a membrane-bound organelle already. Um, um, it has three, is it two or three components? We have fibrillar component and the granular component, yes, fibrillar and granular component. So the fibrillar components include the DNA that is unraveled in chromatin loops plus RNA and RNA component of ribosome. DNA carries genes for rRNA. Then RNA is rRNA. Then that is synthesized and processed. That is, the RNA will be synthesized and processed into rRNA. And it is the dense area where transcription happens. Then the granular components are the proteins, the RNA and the proteins. They are the granular area. They form the granular component. They form ribosomal subunits exported to cytoplasm. And the cell is correlated with cell activity. Okay? So we have during cell division, condensation of chromatin into compact chromosome, then shrinkage and disappearance of nuclei. That is the function of the, like those are the activities that happen in the nucleus, inside the nucleus. The rRNA and protein disperse or get degraded because this time it's just the DNA structure that we need. Then the DNA which has condensed to chromosome, then many tiny nucleoli visible and fuse and become large nucleolus. Then the chromatin or molecular structure of the chromosome. These I've covered in the transcription and this thing. Eukaryotic chromosome, two broad components. We have the nucleic acid and proteins. The DNA, that is the primary nucleic acid, plus small amount of RNA, they transit to the cytoplasm. Then the proteins are just histones that they are basic in their pH, that is their bases, and they are of two types. We have the core histones and the linker histones. Is that okay? The core histones are called the H2A, H2B, H3, and H4. They are the ones that help we talked about the condensation of the chromatin material into chromosome during cell division. So it is the histone that help in the coiling around to condense that chromatin. Then we have the non-histone proteins also. The histones bind to negatively charged DNA and they help to stabilize the DNA. And the mixture of DNA and proteins, that is the basic structure of the unit of chromosome. That is specifically what DNA and proteins. Chromosome contain what? DNA and proteins. Okay, and that's what you will see here. You see, it helps you to condense. This thing that you are seeing like rope on this thing is the DNA. Then that thing inside, this purplish lilac colored or blue colored that you see here are the histone proteins. Okay. Then we have the nucleosome. The nucleosome is the simplest packing structure of the DNA. See, this one structure here is what we call the nucleosome. Do you understand? Called the nucleosome. Now, it contains 146 base pair of DNA wrapped around histone octamer. Exactly. We call it histone octamer because there are um, eight types of those histones. I mentioned H2, H2B. So there are eight. If you count, let's try to count. You see 1, 2, 3, 4, and under also 1, 2, 3, 4. That's eight. So we call them octamers. So octamer are two copies of four. Thank you. Two copies of four core histones. Core histones are just the histones that are within the ribosome, the DNA rather. So the DNA wrapped around one octamer is called the nucleosome. So it is the simplest packing structure of the DNA. Okay, the core DNA are associated with the stone octamer, the linker DNA to DNA between. Okay, that's just, you know. So now we have the model of the packing of chromatin. So remember that chromatin is the DNA structure fully stretched. So you see